This class is on disposal of bio-waste. This is a very important class for you as you will be doing some kind of research work that may generate some kind of bio-waste. This class will explain you about the kinds of waste that we are exposed to in our laboratory environments and how we can dispose of this waste. So the question is biomedical waste different than the bio-waste? Biomedical waste is any waste that is being generated during the diagnosis, treatment, or immunization of human beings or animals or in research activities. This waste is a biohazard, we all must remember. In research at IIT Kharagpur, we generate a large amount of biomedical waste every day in our laboratories, which needs to be disposed of in an environmentally sound way as per Indian law on biomedical waste management. Laboratory environment is similar to hospital environment. This is a very important statement. Please remember this statement since we are at risk of exposure to different kind of samples from humans, human cell lines, animal samples, microbial cultures, molecular biology waste, cytotoxic drugs, and as well as the chemicals and disinfectants. All of these exposures put us at risk of diseases and other health hazards. Now let us see some examples of common laboratory waste. Most important is waste from human and animal origin, such as anatomic waste or pathological waste such as tissues and surgical waste. Please remember, infections can be acquired from all of these wastes. Human blood and body fluids are commonly used in SMST. Body fluids include serum, urine, saliva, cerebrospinal fluid, peritoneal fluid, and other fluids. Please note that body fluids may contain blood-borne pathogens such as HIV, hepatitis B virus, and hepatitis C virus. So these samples are very risky and we all must be very careful in handling these samples. Blood or body fluid contaminated personal protective equipments may also carry blood-borne pathogens, so these must be disposed of in a proper way. Many labs in IIT Kharagpur have cell culture facilities. The cell culture waste is also a biomedical waste coming from research, since human cell lines may contain blood-borne pathogens such as HIV, hepatitis B, and hepatitis C virus. Waste coming out from microbiology research may contain infectious agents. These include culture media plates, liquid broth tubes, discarded cultures, culture stocks, and live vaccines. Now let us study about sharps. Sharps come under a different category of waste. Sharps are substances or objects that can penetrate or puncture skin. These include hypodermic needles, surgical blades, lancets, scalpel blades, Pasteur pipettes, glass vials, ampoules, glass slides, and cover slips. So all of these can puncture or penetrate our skin, and these need to be disposed separately from the solid waste. Molecular biology waste is a common waste in research labs. The first picture here on the left shows an ETHIDIUM bromide containing agarose gel. ETHIDIUM bromide is mutagenic or it can cause cancer. The picture on the right shows polyacrylamide gel. The monomer form of this acrylamide is a neurotoxin. So these gels cannot be disposed of in regular trash. They need a proper disposal method. In addition to the above waste, we are also exposed to drugs, antibiotics, expired medicines, and all these different kinds of chemicals. So with all these wastes near us, we are at so much greater risk of health hazards. This slide shows the hazards from biomedical waste. The most important is infectious agent. Please note that many infectious organisms can survive in environment for longer periods. As you can see, the example of coronavirus being surviving on surfaces. Biomedical waste may also be genotoxic. They may contain sharps, hazardous chemicals, or radioactive substances. This slide shows the common laboratory acquired infections. This topic will be dealt in detail in your last class on case series. I would just say that human cell lines may transmit human T lymphotropic virus that causes T cell leukemia. And blood and body fluids may contain hepatitis B virus. Hepatitis B virus is the major cause of liver cancer, and hepatitis C virus causes liver failure. This slide shows the routes by which we may acquire infections from laboratories. Number one and most important is improper disposal of waste without processing or treatment. Other routes of exposures are aerosolization, splash, contact with contaminated surfaces, sharp injuries or needle stick injuries, working in open bench, not wearing appropriate personal protective equipment, and not maintaining hand hygiene. With the advisories of coronavirus prevention, we all know now that hand hygiene is very important to prevent infections. There is a confusion on who is at risk of laboratory acquired infections. Laboratory acquired infections can happen in those who are directly working with human or animal samples, as well as human cell lines, such as researchers, students, technicians, as well as those who are visiting to hospitals and collecting samples, meeting doctors, and etc. Laboratory members who are in the same vicinity are also at risk of infections. So even if you are not working with human or animal derived samples or human cell lines, and your lab mate is working, but you are also at risk of infection since you are in the same vicinity. Cleaning workers, laundry workers, transport workers who are working with you, and workers in waste disposal facilities, they are also at risk of infections from the waste. Along with these, the birds, rodents, animals, and environment, they are also at risk of these health hazards. Now let us study the biomedical waste situation in India. There was a study in 2002 to 2004 by International Clinical Epidemiology Network, that is INCLIN, where they assessed the biomedical waste situation in more than 350 primary health centers, around 25 secondary health centers, and 24 tertiary health centers. This study was done in over 20 states of India, and in this they found that around 82% of primary centers, 60% of secondary centers, and 54% of tertiary health centers had no credible biomedical waste management system. So this was a very alarming study. In 2009, there was an outbreak of hepatitis B virus in Gujarat. And this was found to be mainly from the use of recycled and use of unsterilized syringes and needles in the population. The first biomedical waste management rules were formed under the Ministry of Environment, Forest and Climate Change by Government of India in 1998, and in 2016 and 18 these rules were amended. The main objective of these rules is to improve the collection, segregation, processing, treatment, and disposal of biomedical wastes in an environmentally sound management. This is a snapshot of Biomedical Waste Management Rules 2016. Regulated biowaste disposal is an essential work practice control. This means that this practice of regulated waste disposal reduces the risk of exposure by altering the way in which it is performed to make it safer. Overall, regulated biowaste disposal prevents infections and diseases from spreading and keeps our earth clean. This slide is very important. This shows the 5 steps of biowaste disposal as recommended by Indian biowaste rules. So we all need to comply with these steps. The first step is segregation of waste. The second step is processing or pre-treatment of waste at the site of generation. So at the site of generation means in the laboratory. The third step is storage of segregated waste at a common site. Fourth is transportation of waste to a disposal site, and last step is final disposal. So the first 3 steps are to be done at IIT itself. I will take you all through all these steps one by one in the next slides. So step 1 is segregation of waste. Biomedical waste is segregated in 4 categories that can be seen in terms of different colors, so that it is easy for us to understand and also to identify. The 4 colors are yellow, red, white, and blue. Here in IIT Kharagpur, we are following so far the yellow, white, and blue colors. Each of these categories are designated for different kinds of waste and have different containers or bags for collection and have different methods for disposal. Now let's study each of these categories one by one. The yellow category. The yellow category is the most relevant, the most common, and most hazardous for all of us who are working in the laboratories and who are handling these wastes. The yellow category wastes include the human anatomic waste, animal anatomic waste, microbiology waste, cell culture waste, biotechnology waste, clinical laboratory wastes, chemical waste, soiled waste, medicines, masks, gowns, discarded linen, mattress, beddings contaminated with blood or body fluid. So all of these wastes come under the yellow category, and these are bagged in yellow bags with a biohazard label. The figure on the right shows all kinds of wastes that come in the yellow category. I would request you to please take a look at this figure carefully if your research falls in any of these categories. The yellow waste, the waste that comes in the yellow category, it is disposed of via incineration mainly, and incineration is basically burning down everything. The other methods of disposal are plasma pyrolysis or deep burial, but incineration is the most common type of disposal for this yellow waste. Here in IIT Kharagpur, we are not doing red category. Red is for recyclable waste, and a major chunk of our research waste is falling under the yellow category, so we will be not dealing with red waste. Coming on directly to white. So what is white waste? Waste sharps, including metal sharps, are disposed in white designated plastic containers. These are called sharps containers. Sharps containers must be made of hard plastic and must be leak proof, and we must always keep this container closed when not in use. The figure on the right shows all the kinds of sharps that will come into this white waste. Please remember that safe disposal of sharps is very important for us, since needle stick injuries with contaminated needles are largely associated with transmission of bloodborne pathogens. The risk of transmission by needle stick injury for hepatitis B is 33%. For hepatitis C, it is 3%, and for HIV, it's around 0.3%. So we need to handle and dispose of these needles very, very carefully to prevent blood-borne diseases. Please remember that when sharps container is almost through 2/3 full, close the container and keep this container in the bio-waste shed for pickup. Sharps are disposed of by sterilization first, by autoclave or dry heat sterilization, followed by shredding or mutilation or encapsulation in the final disposal site. The blue category. All the glasswares, broken glass, discarded glass and contaminated glass come under the blue category of waste. These waste must be disposed in blue dustbins or cardboard boxes with a blue colored marking. Please remember, we cannot dispose glassware in yellow category or in irregular trash. In the final disposal site, the glass is disinfected, or it is autoclaved or microwaved or hydrowaved, and then this glass is recycled. The black waste. The black waste does not come under the category of biomedical waste. Rather, it is for uncontaminated plastic waste, paper waste, or cardboard waste. Please discard uncontaminated waste in black bags or black domestic waste bins. This waste does not need treatment and is disposed as regular trash. Now coming on to step 2. The step 2 is processing or pre-treatment of biomedical waste. This step is done in the laboratory itself, and this is a very crucial step for safe disposal of waste. Laboratory waste is decontaminated mainly by 2 ways. Number 1 is autoclave. You may be using autoclave in your laboratories, but just to brush up, an autoclave is a machine that uses steam under high temperature and pressure to inactivate pathogens. Number 2 is chemical disinfection. Here we use chemical substances to inactivate pathogens. We will study these 2 ways in detail in the next slides. So let's study autoclave. Autoclave literally means automatic locking. It utilizes both high temperature and pressure as the main key features. Please note that as the temperature increases in the autoclave chamber, the pressure also increases inside. These two are directly proportional, and the main objective is to reach the sterilization temperature and pressure. So what happens is high pressure automatically creates a seal to prevent moisture from escaping, and it protects the contents from outside atmosphere. Please remember that autoclave is an engineering control, which is a permanent structure designed to place a barrier between the worker and the hazard. Please don't confuse it with as a work practice control. Rather, it is an engineering control. Now let us briefly study about the phases of sterilization, since we all use autoclave daily in our laboratories. The first phase is the heat or purge phase, where we add water in the tank, and under heat the steam is generated. This steam displaces air within the autoclave chamber. This is called steam displacement. Then as the temperature increases, the pressure also increases in the autoclave chamber until it reaches the sterilization temperature and pressure. The second phase is the sterilization phase, where actual sterilization of your waste or product occurs. For this step, we set a temperature of 121 degrees centigrade, 15 pounds per square inch of pressure for 20 minutes to achieve sterilization. This is a sterilization phase. Now, depending on the type of waste, if it is a liquid waste or solid waste, we can modify the timings of the autoclave cycle. This thing we will study in the later slides. And last step of this phases of sterilization is the exhaust phase, where the steam is removed through exhaust, and after this we can take out our decontaminated waste. Autoclaving works on the principle of moist heat or steam sterilization, which kills microorganisms by protein denaturation. Autoclaving kills majority of the microbes, spores, viruses, fungi, and other organisms. So majority of biological waste is decontaminated by autoclaving. Please note there are also some exceptions such as autoclaving cannot destroy endotoxins and prion proteins. While autoclaving, you please note that whenever you bag your bags for autoclaving, please make sure that steam at very high temperature and pressure must come in contact with every surface or penetrate to the center of an item to achieve sterilization. If you bag the bags too tightly, then steam may not be able to penetrate in the center of the item. So it must be loosely tied, and steam must remain in contact for enough time to sterilize. Unlike dry heat sterilization, steam is more efficient in transferring heat, so autoclaving is more preferred method for decontamination or processing of waste, unlike the dry heat sterilization. This slide shows the types of autoclave cycles for the wall-bound autoclave. These types of autoclaves are usually found in other countries, but still we must know that different kinds of wastes are processed or decontaminated by different kinds of autoclave cycles. So the number one is autoclave cycle, the gravity cycle. It involves fast exhaust and dry phase for drying the item. Hazardous waste is decontaminated by this cycle, and other wastes such as glassware, dry goods, and metals are also sterilized by gravity cycle. The second is liquid cycle, which is as the name suggests, it is used for sterilization of liquids, and this phase has slow exhaust and no dry phase to avoid evaporation of liquids. The third cycle is the vacuum cycle. It is less common. It is similar to the gravity cycle. It just uses the principle of vacuum, and this is used for sterilization of fabrics and animal cages. Here you can see the autoclaves found in IIT Kharagpur. We have manual autoclave and automatic autoclaves. Manual autoclave may be vertical or horizontal. The vertical autoclave you can see in the top right image, and automatic autoclave is in the bottom right. Please remember that depending on the type of waste, you would have to adjust the temperature and time. It's not something fixed. And some labs also have pressure cookers, which also works on the same principle of moist heat sterilization. Now coming on to the second type of waste processing, that is chemical disinfection. As I said earlier, the chemical disinfection involves use of chemical substances to inactivate pathogens. This is a very common method that is practiced daily in our labs for surface disinfection. But please note that these disinfectants must always be prepared fresh, since their activity decreases over time, and they must remain for appropriate contact time for achieving their microbicidal, the virucidal, or fungicidal action. So we want to make sure that these are effective and they kill whatever we want to. And please note that these are chemicals. They may have some health hazards. So prior to use, we must all read the material safety and data sheets for each of these chemicals that we use in our labs. This MSDS sheets, it comes along with the chemical, and you can also check it online. Just add the put the catalog number of that product, that chemical, and then you can read the material safety data sheets. So we all must be reading before use of these chemical disinfectants. The most common chemical disinfectant is alcohol. We all use alcohol daily in our labs. Alcohol is water soluble, that is dissolved in water to make around 70% ethanol solution or 60 to 70% isopropanol solution. Of these, ethanol is mainly used. The mechanism of action of alcohol is protein denaturation and is effective against bacteria, fungi, most viruses including coronavirus and influenza virus. Ethanol is rapidly bactericidal. It is not static. It kills the bacteria, and their cidal activity drops sharply when it is diluted below 50% concentration. So optimum dilution and optimum dilution is required for bactericidal activity. Please remember that ethanol does not kill everything. It cannot kill spores, non-enveloped viruses, and prion proteins. Ethanol is also not able to kill the big three blood-borne pathogens such as hepatitis B virus, hepatitis C virus, and HIV. And isopropanol is also not active against viruses. And if you think that if there is a contaminated needle with blood and you disinfect with ethanol, this ethanol will not be able to kill hepatitis B virus. So we need something else to inactivate these blood-borne pathogens. And please remember that ethanol can catch fire at any time. It is flammable, so please do not use alcohol near when spirit lamps are on. Another chemical disinfectant is sodium hypochlorite solution, which is commonly called as bleach, and bleach is a chlorine disinfectant. Household bleach we commonly use in our households, in bathrooms and kitchen, and this comes with names such as Lysol and Harpic, and these all contain bleach. So bleach is a very powerful disinfectant. It kills everything, and for liquid wastes we use around 5,000 parts per million, around 0.5%, and the contact time is 20 minutes. Please make sure. And for if there is a high organic load in the waste, then please use 1% or 10,000 parts per million. The mechanism of action of bleach is through being a chlorine disinfectant. It has this hypochlorous acid. This chlorine, it has an oxidative action which kills pathogens by breaking the chemical bonds in the molecules of any organism. So it is very highly powerful, and it kills bacteria, fungi, and viruses. It is also able to kill the big three pathogens such as hepatitis B, hepatitis C, and HIV. So if you have any contaminated metal surface that you want to disinfect, please use bleach for disinfecting the blood contaminated or body fluid contaminated metal surfaces. But again, prions are an exception. Prions are very resistant proteins which cannot be killed by bleach solution. There are also some disadvantages. Bleach is a corrosive and to metals. So whenever you are using bleach for disinfecting metal surfaces, please make sure that you wipe down with water, or ethanol after treatment of metal surfaces with bleach, so that your metals, like such as centrifuges, they do not corrode after if you are disinfecting with bleach. And bleach is also an irritant, so it causes irritation to eyes, skin and throat. So whenever you use bleach, please wear appropriate personal protective equipments when you are using bleach. You can use gloves, you can use wear lab coats, and wear eye goggles, safety goggles, so as to protect yourself from bleach. This slide shows the steps for decontamination of liquid waste by bleach solution. Say for example, you are working in a cell culture lab and you have a large amount of liquid waste daily. So we cannot dispose of that liquid waste directly into the sink. We need to disinfect or decontaminate that waste since it is coming out from human or animal cells. So what we do is we make a solution of freshly prepare a solution of bleach. If our stock is 5.25%, we make a dilution of 1:10 in water, which comes around 0.5% or 5,000 parts per million. So if you have a stock of 4%, you have to dilute it to make it to 0.5%, and this 0.5% freshly prepared bleach solution is added to liquid waste in a 1:1 proportion. And we wait for this mixture to sit for around 20 minutes, and after that we pour down this whole decontaminated liquid waste into the sink, and we flush with a lot of water so that it just washes out from the drain. So third step in the disposal of waste is storage of segregated waste at a common site. All biological waste must be discarded in color-coded, respective, segregated and covered garbage cans in a common disposal area. And the figure on the right shows the biohazard waste shed that is in SMST, and we discard our waste in the segregated waste cans over there in this shed. This bio-waste shed is an example of an engineering control. It is a permanent structure that places a barrier between the hazard and you, and all waste is picked up from this area. Step 4 is the transportation of the segregated and processed waste to the final disposal site. The laboratory bio-waste cannot be disposed of anywhere in the environment. It has to be picked up and transported to a designated area by law. And this is done by common biomedical waste treatment facility. These facilities are spread across India. And what these facilities do, they collect and pick up these waste from different hospitals and healthcare facilities, research centers, and they collect and pick up this waste and they take it to the nearest disposal site. In West Bengal, we have in Haldia this facility. So what we do is we set up a contract with like IIT sets up a contract with this facility. These people come twice a week in IIT and they pick up our waste from the SMST and they take it back to the disposal site in Haldia. The last step is the disposal of the waste in the final disposal site. The disposal facility treats the waste based on the type of waste. For yellow waste, it is mainly treated by incineration. 90% of biomedical waste is incinerated. So all the waste, it is converted to either gas or ash, and other methods are deep burial or plasma pyrolysis, but incineration is mainly used. For sharps waste, what they do is they do autoclave followed by shredding or mutilation or encapsulation, and then they send it to sharp pits or iron foundries or to landfills. The blue waste, that is glass waste. The glass waste is disinfected, or it is autoclaved or microwaved or hydrowaved, and then these are recycled. Now I would go over with the 8 steps for disposal of laboratory waste that is being followed in SMST. So first step is we need an autoclavable, puncture proof and tear resistant bag. So any plastic bag cannot be used for disposal of waste. We need an autoclavable and a tear resistant bag. The second step is we need to fill the bags only when they are 2/3 full. If we do it more, then during autoclaving the bag may burst and the contents may come out. Now the third step is we put a loosely put a rubber band around the neck of the bag so that the steam can penetrate the bag when we are doing autoclave, because we want to make sure that steam penetrates to the center of the item to achieve sterilization, because we want everything to be sterilized. Now you place this bag in an autoclavable tray, and if you have an autoclave tape, please put that tape on the side of bag. This will make sure that your autoclaving is right. And then we put this bag into the inside the autoclave. Now we need to set the temperature and timings. So sterilization temperature and pressure will remain same of 121 degrees centigrade and 15 pounds per square inch, but the timing may vary with the kind of waste. For solids you do sterilization for at least 30 minutes, and for liquids you do sterilization for 45 minutes. So step 6 is once your autoclave is done, you make sure that its temperature goes down, it has cooled down, and you wear a cryo glove and you take out your biohazard bag that is autoclaved. Now in the case of liquid wastes, what you do is, if it is a harmless waste, it doesn't have any harmful chemical, then you just discard this autoclaved liquid waste through drain or sewer with large volumes of water. And if your liquid waste contains a hazardous chemical, then you need to follow the instructions of that chemical on how to dispose of that chemical liquid waste. Now the seventh step, that is in which what we do is this, you take this autoclaved bag and you place it in the yellow bag which has a biohazard label. So this bag is provided by this bio waste management company that comes here to IIT Kharagpur. So we put our autoclaved bag in this yellow bag and we seal the bag. So this sealing or tying of the bag is very important for disposal. Otherwise these people will not take this open bag. It has to be tied. And then you take this tied yellow bag to the garbage can in the biohazard waste shed, and then you are done with your steps. And then these bio waste disposal company, they will come and pick up your segregated waste to the final disposal site. Now a few considerations while you are autoclaving liquid wastes. Please make sure when you are autoclaving liquid waste, it must be in borosilicate or Pyrex glass, that is heat resistant glass containers, and make sure that these containers, they are not filled more than 2/3 of their capacity. And you loosen the caps or you use vented closures so that steam can penetrate inside your liquid and it can sterilize each and every content of the liquid waste. And whenever you are doing autoclaving, make sure that your contents do not have any kind of solvents or flammable or volatile or corrosive liquids such as ethanol, phenol, trizol, chloroform, bleach, and ETHIDIUM bromide, because you do not want to have any blast inside the autoclave. It can be very corrosive, and it can create a blast inside the autoclave. And if your material contains any ammonia or strong acids, please do not add bleach to these materials. Some of us do laboratory animal research, and to dispose of animal carcass or animal anatomic waste or animal body parts, We cannot autoclave these waste. What we do is we store these carcass or body part in the yellow bags, and we keep it in freezer to reduce putrefaction or decomposition. And then we set up a time with this waste management company, and whenever they come to IIT, you just hand over this bag to them, and then they will take it to the disposal facility for incineration. So please do not autoclave the animal carcass. Now I'll go over with the disposal of two kinds of gels that are being used in molecular biology. One is ethidium bromide containing gels, and another is polyacrylamide gels. So ethidium bromide is a dye that can bind to DNA, and it is commonly used in molecular biology research in many of the labs in IIT Kharagpur. Ethidium bromide is a very strong mutagen. That means it can bind to DNA, and it can also lead to cancer. So we have to be very careful while handling this chemical. It can be readily absorbed through the skin and is very highly toxic through inhalation and eyes. So whenever you handle ethidium bromide while making your gels or while making your agarose gels, please wear personal protective equipment such as gloves, safety goggles, and lab coat. Ethidium bromide waste that will come out after your gel is run, it should not be poured down the drain or thrown in the trash unless the waste has been deactivated or filtered. So if you have ethidium bromide powder in your lab, or you have a ethidium bromide stock solution, and you have ethidium bromide gels in your lab, or you have solutions with concentration of at least more than equal to 0.1% of ethidium bromide, and you have contaminated gloves or solids that came in contact with ethidium bromide, or you have spent charcoal filters or destaining bags that were used for filtration of ethidium bromide, then all of these categories, they have high amounts of ethidium bromide, and this cannot be thrown in the regular trash. Rather, this must be contained securely in sealed plastic bags placed in a rigid container that has a lid, and this must be disposed of to an authorized cytotoxic waste incinerator. Now, for right now, I am not sure whether we have a cytotoxic waste incinerator contract with IIT, but at least since the yellow category of waste, it also includes the cytotoxic waste. So I think the ethidium, all of these categories above, we can put all of these in the yellow bag, and then we can hand it over to this waste management company for incineration. We run these ethidium bromide gels in buffers, and these spent buffers after run, they contain ethidium bromide. So this is ethidium bromide liquid waste that we need to do and that we need to handle. So this liquid waste we cannot dispose of in sink directly. This liquid waste should be collected in a labeled glass bottle, and this must be treated with ethidium bromide filters which can bind and remove ethidium bromide. And there are few ways how we can remove ethidium bromide from this liquid waste. Number one is activated charcoal, which binds to the ethidium bromide. Another is destaining bag, as you can see on the right bottom, that you put this green bag into the ethidium bromide waste solution and you leave it overnight, and this destaining bag will bind and remove the ethidium bromide, and the liquid will be clear next morning. And another conventional method is the chemical deactivation method, that is Lunn and Sanson method. And following deactivation, you just pour the filtrate down the drain under copious amounts of water. Now, the disposal of polyacrylamide gels. Acrylamide is very harmful. It is known to cause cancer among laboratory animals. It is a potent neurotoxin, and it is toxic by all routes of exposure and has long prolonged effects. The monomer of acrylamide, that is bisacrylamide, is dangerous because the powder dust can easily become airborne and it can enter the respiratory system. So whenever you are handling acrylamide and making some solutions of bisacrylamide, use a chemical fume hood to measure acrylamide into pre-weighted and closable containers so that you are not exposed to this harmful chemical. And while you are doing all of these operations, please wear gloves, a lab coat and goggles. And when you are adding STS powder, please wear dust mask when you are handling STS. Unpolymerized acrylamide in the gels is toxic and hazardous, so polyacrylamide gels must be disposed in a leak-proof sealed plastic bag, and you place this bag in a labeled cardboard box. You label it as polyacrylamide gel, and then you dispose it as a hazardous chemical waste. But do not put a label of biohazard on the polyacrylamide gel box. These are some general laboratory waste disposal procedures that we must follow while working. So plasticware from tissue culture lab must be treated with sodium hypochlorite solutions before being drained and autoclaved. Ice bricks should be stored in the freezer for future use or thawed out or dried off and put in a black bin bag. Dry ice should be allowed to sublime to gaseous form, ideally in a fume hood. All empty solvent bottles need to be put in the fume hood to evaporate before washing. Please make sure that there is no liquid left in empty glass or plastic bottles. So what is not right here in this slide? There are a couple of pictures here that have some non-compliance issues, and if we go, I'll go through this picture step by step. First we'll go through the top row, and first image of the top row on the top top left is we have a red biohazard bag which is not sealed. The waste is outside the bag, and all this waste is left in the open. It is not in a biohazard waste can, so we cannot leave the biohazard waste out in the open. Now if we move to the second image, we have a green bin, and we can see that there is a red bag on the sticking to the side of the wall of the bin, and there is waste, loose waste in the bin. So we cannot discard waste in an untied bag. We need to tie the bag and then we discard the waste. Now the third picture we can see is that there are two bags that are stuffed very tightly in a tray that is not able to accommodate these two bags. So whenever you do autoclave, you put bags in such a way that that steam can penetrate and they are not overstuffed while you are autoclaving, because you want each and every area to have the access of steam. Now the rightmost picture is we have this biohazard waste bin that has a blue bag, a white bag, some loose stuff. So this is absolutely not required. This is not allowed, and we need to throw only the designated biohazard waste bags. It has to be yellow bag, and the waste has to be tied in within the bag. We cannot discard in as loosely, and we cannot discard in any other plastic bag. So it has to be a biohazard bag. Now if we move to the bottom row, on the left hand side there is a bag that is totally tied to the very top of the bag. Remember, we need to tie the bag only so when they are 2/3 full. We do not want to tie it up to the top. Now the second one in the bottom row is here we have these three bins, and again these are over full. This has to be 2/3 full is the limit that is required for biohazard bags. Now the last image here in the bottom row. So I would like to thank you here for your patient listening. And the main point about this whole presentation is that we cannot dispose of anything and everything out in open that is coming out from our labs. And so there has to be a regulated protocol that we all need to comply, and this will help ensure protect our lives as well as others' lives, and it will help keep our environment clean, our earth clean for others to live. Thank you.